Hi everyone, my name is Thandi Nhleko, and today I'll be presenting on hemoglobin, the structure and functions of hemoglobin. Now, what is hemoglobin? Hemoglobin is the iron-containing coloring matter of the red blood cells. It is a chromoprotein forming 95% of dry weight and 30 to 34% of wet weight. The function of the hemoglobin is to carry respiratory gases, oxygen and carbon dioxide. It also acts as a buffer. Molecular weight of the hemoglobin is like around 68,000. Here I have a near structure of hemoglobin, and as we can see, here is the red blood cell, and here it is an opening that shows us the structures and how it binds with oxygen. As I had said, oxygen binds with the hem of the molecule. What are the functions of hemoglobin? The functions of hemoglobin is to transport respiratory gases. So that means it's oxygen from lungs to tissues, carbon dioxide from tissues to lungs. The first one, transport of oxygen. When oxygen binds with hemoglobin, a physical process called oxygenation occurs, resulting in the formation of oxyhemoglobin. The iron remains in the ferrous state in this compound. Oxyhemoglobin is an unstable compound and the combination is irreversible. When more oxygen is available, it combines with hemoglobin, and whenever oxygen is required, hemoglobin can release oxygen readily. Secondly, it assists in the transporting of carbon dioxide. When carbon dioxide binds with hemoglobin, carb hemoglobin is formed. It is also an unstable compound, and the combination is reversible. The carbon dioxide can be released from this compound. The affinity of hemoglobin of carbon dioxide is 20 times more than that of oxygen. It also acts as a buffer action. Hemoglobin acts as a buffer and plays an important role in acid-base balance. Moving on to the structure of hemoglobin. Hemoglobin is a conjugated protein. It consists of a protein combined with iron containing pigments. The protein part is the globin and the iron part is the hem. The hem also forms a part of the structure of myoglobin and the neuroglobin. Moving on to the iron. The iron is normally presented as the Fe2+. It is in an unstable or loose form. In abnormal conditions, the iron is converted to ferric state, which is a stable form. Moving to porphyrin. Porphyrin is the pigment part of the hem. It is formed by 4 pyrrole rings called I, 2, 3, and 4. The pyrrole rings are attached to one another by methane bridges. The iron is attached to nitrogen of each pyrrole ring and nitrogen of globin of each molecule ring. The globin contains of 4 polypeptide chains. Among the 4 polypeptide chains, 2 chains are alpha chains. Moving on to the types of hemoglobin. Normal. We have 2 types of normal hemoglobin, and these are adult hemoglobin and fetal hemoglobin. Replacements of fetal hemoglobin by adult hemoglobin starts immediately after birth. It is completed at about the 10th of age. In adult hemoglobin, the globin contains 2 alpha chains and 2 beta chains. In fetal hemoglobin, there are 2 alpha chains and 2 Y chains instead of B chains. Functional difference. The fetal hemoglobin has more affinity for oxygen than the adult hemoglobin, and the oxygen hemoglobin dissociation curves of the fetal is shifted to the right. Now I will move to the disorders of hemoglobin. Abnormal types of hemoglobin are a variety, and these are constituents because of pathological reasons. The first one could be most of the mutations do not produce any serious problems. Occasionally, few mutations result in some disorders. There are two categories of abnormal hemoglobin. Firstly is hemoglobin pediasis, and two is hemoglobin in thalassemia and related disorders. Thank you.